Hey, Jenny here, and I'm pleased to be visiting today with Dr. Alexandra Belayew, who with great humor says that she is the unproud mother of DUX4. It is she and her lab that are the ones who discovered DUX4. So we're going to learn more about that here in a few minutes. Welcome, Dr. Belayew.
Well, thank you very much for the invitation, Jenny.
You're welcome. So as patients, many times, if not always, we feel like research moves slowly, but at the same time, we know that it's a difficult process. And so it's my understanding that the DUX4 story was a difficult one for you to get out there, which is key for identifying the right therapeutic target. Can you tell me a little bit about that DUX4 story, that journey that you started and have continued?
Okay, so that's a saga, I would say.
Right.
So the starting point is that at the time I was not yet at the University of Mons. I was at the University of Leuven, and I was working in a very large laboratories. And when I had joined the lab, the big boss, Professor Désiré Collen, told me there is a very bright Chinese MD who would like to join the lab to make a PhD. And he wants to work in molecular biology. Alexandra, I've hired you because you were expert in molecular biology. Would you take care of this young man? And this young man is Hao Ding. I probably don't pronounce correctly for Chinese. I do apologize. And so Hao Ding uh, is a very nice guy, was already at the time. Mm-hmm. And he started working, and this guy had some behavior as a young researcher that every laboratory head would dream of. That is, if I would come with a new idea because I had heard at a meeting or I had read a scientific paper, and I said, well, that would be interesting. He said, okay, I want to do it. While most researchers would say, oh, it's looking complicated, I'm not sure I'm going to be able to fit that in my very tight schedule. And while Hao Ding would immediately say, okay, let me do it. And he would come and work on weekends. And when I arrived in the lab on Monday morning, he would come with all the data he had harvested since Friday evening. And it would usually take me 2 to 3 days to sort out the meaning of what he had found. So we were really, he was working like a pioneer. He would do sort of standard experiments, but with the DUX4 story, they would never give the expected answer. And it was very stimulating from an intellectual perspective, but at the same time, very frustrating that you could never say in advance what might happen. So the basic experiment he has done was, we had it was already in the lab before I arrived. I had just joined the lab. There was a new transcription factor that had been identified. So what is a transcription factor? So you know that we have all the information to make our cells and our body is DNA. And DNA is organized in genes, in the chromosomes, which are in the cell nucleus. And so the chromosomes contain the genes, and each gene is a recipe to make a protein. And we have about 20,000 genes, we humans. And obviously, all those 20,000 genes are not working in every cell at the same time. You have different genes working to make a cell of your skin, then genes making a cell of a muscle, then genes making cells in your brain. So there must be some kind of organization in the cells which decide which gene is going to be used now in this cell. And what is deciding this is a collection of special proteins, which are sort of chefs, which are deciding which gene has to be switched on, which gene has to be switched off at any moment in any cell type. And so we had in our hands one transcription factor that had been isolated by somebody who had left the lab. And the question we were wondering is, this transcription factor, this chef, what genes is it acting on? And usually a transcription factor can act on several hundred different genes. And so it's always interesting to know what are the genes which are activated by this factor. It helps you understand what is the role of that factor in the body. And so Howding told me, okay, if you want, I could go fishing for the genes onto which this transcription factor is binding and which it can activate. And so he went do his experiment. And at the time, it was really pioneering work. No, very few people had done that already. Now, if you want to do that, you have kits that you can buy, and that will make life much easier for you. But at the time, it was all, things you had to prepare by yourself. So one Monday morning, Hao comes to me and says, okay, I have fished out a number of pieces of gene, and I have read their sequence, so the order in which the letters A, T, G, C are on the DNA. And I can give them to you, and maybe you can find where they come from, these little pieces of DNA. And these were the early days of working with computers on the human genome sequence. So the human genome is like 3 billion letters. And at the time, there was maybe 20% known. Now it's all known. It's part of, it's like the dictionary of life that every researcher has available. But at the time, only part of it was known. And so I went to my computer and started searching. And among the different genes that I identified, so I said, okay, this piece comes from that gene, that piece comes from that other gene. And then suddenly there was this name I never heard of before, D4Z4. And I thought, what is that D4Z4? And not being myself a geneticist, I didn't know that this nomenclature D4Z4 is typical for repeated elements. And so I was a little, I could not understand exactly what was going on. And I asked to the next door lab where they had very bright geneticists. And I said, could you please explain what is that? And the guys explained to me, well, it's a repeated element that's located on chromosome 4, very close to the end of one arm of chromosome 4. And it has been related to a disorder which is named FSHD. I had never heard of FSHD. Nobody has ever heard of FSHD. Right. And so there we were a little stuck, because even this geneticist had never heard of FSHD either. So then we asked to somebody else in the group, who was a graduate student who was Flemish. So the University of Leuven is in the part of Belgium where people speak Flemish. So this young Flemish guy had the basic training as a bioengineer. So he knew how to grow crops and sell milk on a large scale and things like that. But he had studied molecular biology. He liked that very much. And so he had just joined the group. And in fact, the big boss of the lab was not too happy with him. One day he had told me, you know, this guy, he spends too much time on his computer. He should be more frequently at the bench. And so I told to Desiree Collen, well, listen, this guy, he can organize his work as he likes. It's no problem for me. And so I went to this guy. His guy is named Jan Gabriels. And so I asked Jan, you are an expert now at internet, which was only developing at the time. So Google was not there yet. And so, Jan, can you find some information about that disease, FSHD? What is it? And so Jan searched on his computer in the internet, and he found out the FSH Society, which has now a new name. It's FSHD Society, but at the time it was named FSH Society. And this society was headed by a woman, Carol Perez, who was the mother of Daniel Perez, the recent past president of the FSHD Society. And so he told me, Jan told me there's plenty of information on FSHD there. You know, it's a muscle disorder, and it's a dominant transmission. So if one of the parents is affected, one out of two children will statistically get it. And nobody knows what gene is causing it, but it has been linked to a change in copy number of this D4Z4 repeated element. But people have no idea where the gene is. And they know there is no gene inside of the D4Z4 unit. And so they're looking around it, thinking if you change the number of copies, maybe you are affecting activity of a gene located a little bit to the right or a little bit to the left. And this is probably where one should expect to find the FSHD gene. So there we are. We are looking for the beginning of a gene with this transcription factor, and we fish out something in the middle of a D4Z4 unit. And the test we did then was to see whether this piece of gene was active. A gene has a beginning, which is named the promoter. It has the recipe to make a protein, and it should be an AND, which is named the polyase signal. And so what we had found was a promoter. So we then looked inside of the D4Z4 unit and found that there was a recipe to make a protein next to this promoter. So we thought this looks like a real gene. But it was very strange because it had been considered since the 70s that repeated elements in DNA were sort of stumbling on words, you know, that stuttering, yeah, that were meaningless. And so nobody believed there could be a gene there. So we came with a bright idea. We have a promoter that is active. We have the recipe to make a protein. And so what? And it took us several years to be able to demonstrate that this was a real gene. How can you show that the gene is active? The gene, before it makes the protein, the gene has to copy it to make the RNA, the messenger RNA. And this messenger RNA will leave the cell nucleus and be used to make the protein. And so to demonstrate that the gene is functional, you have to be able to detect the messenger RNA, and you have to be able to detect the protein. Mm-hmm. And it was extremely difficult. Even today, it is still very difficult to detect the DUX4 protein because it's produced in such small amounts that it's really tricky to be able to find it. And we didn't have the tools to find a protein. And the standard method to detect the RNA did not work for DUX4 because not only do you have those repeated elements, D4Z4 on chromosome 4, but you have hundreds of repeated elements on other chromosomes, which are extremely similar to D4Z4. And all those repeated elements are also making RNAs. So that when you want to isolate the RNA that's coming from D4Z4, from this new DUX4 gene we had found, you have to sort it out from the hundreds of other RNA that looks extremely like itself. So it was really finding a needle in a haystack.
Mm-hmm.
It was very difficult. So these were the beginnings.
Mm-hmm.
And basically it took us many years to be able to demonstrate that. And we had several problems besides the technical problem of detecting very low abundance protein or RNA lost among hundreds of very similar ones. There was a problem that we had not understood. We had made an artificial gene that contained a recipe to make DUX4. And like is done usually in labs, we took this gene and put it in cells that were kept in culture in plastic dishes in the lab. So muscle cells that we could buy from various companies. And we put in there the DUX4 gene and see whether we could produce a large amount of the protein that we could try to detect. But every young student in my group who had tried to do that experiment came extremely depressed, telling me, it's very strange, I have only a few cells left after I put the DUX4 gene in them. Oh. I don't know what happens, but I sort of lose my cells when I put DUX4 in there. And so the immediate reaction of a young scientist who is in training when an experiment is failing is to wonder whether he has not done something wrong. So those poor kids were just, destroying themselves by saying, what might have I done wrong to these cells that they're not there anymore? And we only got the explanation at a meeting organized by the FSH Society, because Daniel Perez very kindly invited us to join the meetings. And there we met Alberto Rosa from Argentina. And Alberto had also thought there might be something interesting in the D4Z4 elements, but just by looking at the sequence. And so he had also tried to produce the protein, but the sequence that was available had mistakes in it, so it was very complicated. And he came with the same problem, that the cells seem to disappear when you put the DUX4 gene in them. And he came with the very logical conclusion. This gene produces a toxic protein. And so when you put the DUX4 gene in a cell, the DUX4 protein is killing the cell. So if you wait too long, you don't have any cells anymore in your dish. So you really have to look very quickly, like 16 hours after you have put the gene in the cells, to be able to detect the DUX4 protein. If you wait longer, everything is dead and destroyed. And it was Alberto Rosa who came with the explanation. And we looked at each other in my group and so we were so stupid. I mean, we had that in front of our eyes and we never thought this could be such a dramatic toxicity, because it was destroying all the cells. In 24 hours, there was nothing left in our Petri dish. So, yeah, that's the point. And so we collaborated with Alberto to publish those results that DUX4 was inducing cell death when we were expressing it in cells.
And that's apoptosis, right?
Yes. Okay. So right. It's very nasty because it's sort of pushing the cells to commit suicide.
Right.
That's what apoptosis means. You know of apoptosis as a natural process when you walk in a forest at fall, that all the leaves are falling down. Right. Apoptosis occurs just at the tip of the leaf where it's attached to the tree. Right. Those cells die, they commit suicide, and so the leaf is not holding to the tree anymore and falls down. Mm-hmm. So apoptosis is something that is programmed in our genes and in the genes of plants and whatever animal. It seems DUX4 is triggering that process in muscle cells.
And so you've got this all figured out and you're ready to tell the world but the world was not ready to hear that. Tell us a little bit about that. Was was it hard? Is a hard sell to say DUX4 is what we're looking at?
Yeah. That was tricky. So the difficulty was really between the time we had identified the DUX4 gene and the time that Alberto helped us to understand that it was toxic. That period of time was very difficult. And because we had to develop all the tools to allow detection of the RNA and to allow detection of the protein. And it took us, I think, 5-6 years before we got all the tools, because every standard method we would try to use was not working. One difficulty is that the gene, and thus the RNA, is very rich in G and C letters. And you have 4 letters, A, T, G, C. And when you have a lot of GC, the RNA and the DNA is very sticky. And it makes all the ways you handle it, or the enzymes handle it, extremely difficult. And so we had to sort out exactly what method. And there were publications that were coming out because we had published already that we had found the DUX4 gene in D4Z4. And several laboratories tried to detect the RNA and could not either. So we were not the only stupid at the time, in a way.
Right. The whole group.
But these guys, they were publishing, and they were publishing that, listen, those people from Mons, because at the time I had moved to Mons, they're telling you anything it's completely unreal. There is no such thing as an active gene there. We cannot find the RNA, so there is no gene there. We cannot find a protein, so there is no gene there. And so it was really tough. And finally, we were able to find the full messenger RNA. And most of it was inside of the D4Z4 unit. So that was the part that was copied hundreds of times over the genome. But then, very surprisingly, and it was a very, how would I say, calm and systematic and very gifted molecular biology in my team who found that, Eugénie Anceau, she realized that the end of the DUX4 RNA was outside of the D4Z4 unit. And this was completely unexpected. Which is stupid. I mean, we scientists were seeing a pattern, D4Z4 repeated, so everything had to be inside of that pattern. But the enzymes that copy the DUX4 gene, they don't see the pattern, they don't care, they copy the gene as long as they are allowed to do so. And so they could copy outside of the D4Z4 unit, and there they were meeting the end of the gene, which is the poly-A addition signal that you have heard of a lot, because this is required to develop the pathology. You must have this signal. So what does it mean? It means it's a signal that says that at the end of the messenger RNA, there will be a stretch of A's that will be linked to each other. And this poly-A, so several A's, this poly-A tail is something that is protecting the RNA from destruction. And when a messenger RNA has a poly-A tail, it can survive for a longer period of time inside of the cell. And the recipe it carries to make a protein can be used, and many proteins can be produced from a single RNA with a poly-A tail. And so Eugénie found the end of the gene, Houding had found the beginning, and the recipe I did not mention had been found earlier by Jane Hewitt, who was the first person who had sequenced the D4Z4 repeated elements. But she had not found a gene, she had not found a promoter, so she thought it was junk DNA. And to put together, so the promoter from Houding, the recipe from Jane Hewitt, and the end from Eugénie Anceau, we had a real full gene, a full RNA. And we were intrigued by this discovery, and we mentioned in the paper that we published about that that the presence of this poly addition signal was probably something that was very important to express the DUX4 protein. And so that was our conclusion at the time. And we could not publish what we had, so we had found the RNA. We were able at the time to detect the protein, and in muscle biopsies, we could homogenize the muscle biopsies and analyze them and see the DUX4 protein. It was extremely difficult, but we were able to detect it. But we could not publish that. The journals would not accept this. It was not enough because we did not have a function for this protein. How can we say, so it's nice, we have a gene, the RNA, the protein, but so why is it causing the disease? And then at another meeting of the FSHD Society, we would go there every year, we met Yiwen Chen. And Yiwen Chen is a bright scientist working in Children's Hospital. It's not the name, I forgot the official name of her foundation. It's in Washington. And she is an expert of RNA analysis. And what she had done, she had taken muscle biopsies from 10 different disorders, and muscle biopsies from people who were not affected with a muscle disorder. She had extracted all the RNAs and sequenced them all. And so for each muscle biopsy, she had thousands and thousands of RNA sequences, and she compared them with a computer to find what was special in FSHD muscle biopsy as compared to the other disorders and to the non-affected muscle. And she came out with one RNA that was more abundant in FSHD biopsies. It was not DUX4 because DUX4 was so low abundance that she did not even see it, but it was PITX1. And PITX1 was very interesting because it was known already at the time that PITX1 was also a transcription factor. So the protein could give orders to genes to make the lower part of the body when an embryo is developing. And it was also known that when you were disturbing the level of PITX1, it could cause a muscle atrophy, so muscle shrinking. And so she was very excited with that. And she thought, well, I have the FSHD gene, it's PITX1. But the only problem is that the PITX1 gene was not on chromosome 4. Okay. And it was established that FSHD was linked to chromosome 4. So she was coming with another chromosome, and nobody would publish her data either. So at this meeting of the FSHD Society, she presented a poster, and I was intrigued that PITX1 has a number of aspects which are similar to DUX4. And these are genes which are named, which have a structure named homeobox. And the homeobox is typical of genes which are involved in embryo development. And it is known that, for instance, to make an eye, you need one homeobox protein named PAX6. And with just activating this gene, you produce a chef which can build an eye. So these transcription factors are extremely potent. And so it was nice that PITX1 that she was finding expressed very much there was in the same family as DUX4 that we thought was causing FSHD. So I went to see Yiwen and I said, listen, I think your gene is activated by my protein. So that DUX4 is probably activating the PITX1 gene. Do you want to test that? Because if this is the case, then we will be saved because we will have a function for our DUX4 protein and a link to the disorder because she has found that PITX1 was expressed in large amount in the muscle biopsy. And so we went back home to our respective labs, and she had a graduate student who did the experiment, and I coached the graduate student online. There was no Zoom at the time, but there was Skype. Right. And so she did the experiment and came to that conclusion that this was the case. So we put all our data together and we submitted to a journal of the American Academy of Science, and it was accepted. But it was very hard. And you know, when scientists submit a manuscript to a journal to get it accepted for publication, the journal will send the manuscript to experts in the field. And those experts will read carefully what you have written in your manuscript and decide whether your experiments were done okay, or whether you were missing some points, or whether you had a wrong interpretation of your data. And so the experts, the reviewers, will send the set of questions to the authors, and the authors have, I don't know, 1-2 months to answer maybe to those criticisms, sometimes have experiments to do again, and then submit, correct the manuscript, submit it again, and then the editor evaluates whether you have correctly answered to the reviewer's questions. And one of the questions was very intriguing. It was, several papers have been published which could not detect DUX4 RNA. So please give every detail on how you proceeded and compare every detail to the ones that these people used who failed. And so we made a big table, which is at the end of the paper, where we say, Mr. Such and such is working at 37 degrees. We thought it was better to work at 25 for that step. And he used so many % of salt in his buffer, and we thought it was better to put less, and so on and so on. He ordered this enzyme from that company. We considered the enzyme from this other company works better. And it's really like, you know, cooking tips right, to demonstrate how to do. And because we published that table, then now anybody was able to detect DUX4. And that's when big labs, which were experts in other fields, started to join in the FSHD field, like Stephen Tapscott, for instance, used our method to detect the RNA for the first time. And so that was a relief to see that other people could reproduce independently from us the same data. Not really independently because they use the same technology, but at least we were not the only crazy in the FSHD story. Other ones were seeing the same DUX4 RNA and the same DUX4 protein. So people started developing their own tools that they validated with our tools, and then they could start working on their own. But it was really the key point was this publication. So the 2 papers came in a few months distance. So the one with Alberto Rosa showing that DUX4 was toxic, and the one with Yuan Chen showing the full RNA with the end outside of D4Z4, and the fact that the DUX4 protein could activate the PITX1 gene that whose RNA Yuan Chen was finding much more abundant in FSHD biopsies. So we had a whole story there.
That's quite a story. That's quite a journey for sure. So,
yes, I'm sorry. I've probably lost most of you on the way in my explanation, but oh well.
Very thorough. That's right. So during the point where the papers were published, then researchers, then you mentioned, could reproduce the same functions in their labs. So up until that point, though, you know, there's probably a lot of research going in the wrong direction because they didn't have details about DUX4, right? So
that's right.
So that,
and what I found very depressing is that when we were going to those meetings of the FSH Society every year, it was somewhere in October, we would go to the United States and go to one of those meetings, and every year we would see new young researchers starting a project about DUX4 and turning around that. And the following year, these guys had disappeared.
Okay.
They had not been able to develop the tools to do that study, and they had this toxicity problem which they were not aware of yet. And in the life of a scientist, if you are unable to publish regularly, then you progressively lose funding, and so you start, making pizzas or
Yeah.
whatever else, but you cannot do research anymore. So I'm sure that a number of bright scientists who tried to join the FSHD field at the time were completely disgusted, disappointed, and were lost to science, or at least to science of FSHD. And it's really a pity, because so much energy and frustration is terrible. But now that we had this paper, and PNAS, the journal where we published the proceedings of the National Academy of Science, is a well recognized journal. And so that gave some weight to what we had been claiming. And so people could start trusting that. And so that now people had something that they could see on the horizon, which was DUX4. It was, but perhaps still a very long way to get there and to understand what it was doing, but at least the target was set.